We meet in an hour of change and challenge, in a decade of hope and fear, in an age of both knowledge and ignorance. The greater our knowledge increases, the greater our ignorance unfolds.
What is synthetic biology? Synthetic biology is a process for designing and building or engineering living organisms.
They won't be living creatures like we know them to be. We'll have, yes, we're entering an age where we are constructing devices, machines that violate all our categories.
Overall, our best defense against biological risks is the speedy advance and dissemination of the life sciences.
Scientists will think the research is really good, it's reasonable, it's exciting, controllable, right? Because you're smart and it's cool. Academics like me and activists like ETC people will think that it's maybe not, maybe it's unsafe or too capitalist or it's unfair or it's insecure or just basically repugnant. And actual people are going to love it.
Right now, synthetic biology is at an early stage.
Philosophers worry that this will be the thing that changes human nature.
In biology, we have the best technology for manufacturing that human beings have ever encountered. Let's learn how to use it in a way that's constructive and responsible.
But again and again, we've been confronted with significant scientific advances.
Mechanical engineering and structural engineering and electrical engineering and environmental engineering and chemical engineering and the history of technology.
And we still putter along like ordinary human primates, surprisingly similarly throughout the changes of science and technology.
This technology is going to bring huge humanitarian benefits. How can we advance this technology safely and securely? How can we disseminate it so that people benefit from it?
Biology is this amazing technology for manufacturing, for making things, right? So trees, people, plants, right? Like almost everything we love is manufactured by biology. Yet we're so inept at using it to construct things in ways that are useful and constructive.
This is Drew Endy.
Yeah, let's go for it.
And he is leading the charge to harness the genetic code for building living machines in what has been termed synthetic biology.
So synthetic biology is pretty interesting.
And that's me, at the beginning of my journey to understand just how synthetic biology represents a new paradigm for scientific research.
So I'm on my way to Washington, D.C. to meet with Lori Zoloff, professor of medical ethics and humanities at Northwestern. She also heads their Center for Bioethics, Science, and Society. I'm catching up with her at a stem cell conference, but she is also at the center of the discussion about the ethical implications of synthetic biology, and she really helps put it in a historical and philosophical context.
The important thing about synthetic biology isn't the synthetic as in fake, it's the synthetic as in synthesize, as in coming together, and that's as interwoven. That's the field, that's the part of the field that captures the interests of moral philosophers, is the capacity to really reintegrate fields of knowledge centered around a goal or a purpose rather than a separate intellectual discipline.
Our obligations to ourselves, as well as others, all require us to make this effort, to solve these mysteries.
It begins here, where the genetic code programs the workings of the cell.
30 years ago, recombinant DNA technology marked the beginning of the modern era of biotechnology. Folks were able to use restriction enzymes and DNA ligase to cut and paste together pre-existing fragments of DNA.
In this way, using restriction and ligation enzymes, you insert a gene of your choosing into a foreign environment, where it will be expressed and replicated. And this technique is the bedrock of genetic engineering as we know it. Enhanced agriculture is just one of the things that recombinant DNA has brought us. Others include synthetic human insulin and fish that glow in the dark.
After recombinant DNA came polymerase chain reaction and then sequencing of DNA. And these three technologies let us write and read out genetic material. Well, synthetic biology says that's fine. We've had those tools for 30 years. The field of biotechnology is sort of a young adult just reaching its potential. But is this all there is? Could we get much better? What new tools and technologies might one like to see?
What would it mean to bring an engineering discipline to biology? Drew Endy identifies three essential elements, the first of which involves synthesis of DNA from scratch.
So automatic construction of DNA based on DNA synthesis, which lets individuals push a button, send in a sequence of DNA to a computer, and get that DNA printed from scratch. You can have 4 bottles. Here's a bottle of T, here's a bottle of A, here's a bottle of G, and here's a bottle of C. And the chemistry here is decades old, 26 years old. And so what's interesting today is that for the last 10 years, people have begun to make investments in the technology of DNA synthesis, the process engineering, the machines that take these chemicals and print them to make synthetic DNA. So the technology for taking these chemicals and synthesizing DNA over the last 10 years has been improving by about a factor of 2 every 12 to 18 months, which means it's improving faster than computers have been getting better.
The next tool involves creating an abstraction hierarchy.
TAA, TA, CGA, CTC, ACT, ATA, GGG, AGA is a consensus promoter sequence for the T7 RNA polymerase, a 23 base pair piece of DNA that initiates transcription, or the beginning of gene expression. It's incredibly tedious to memorize such sequences of DNA, and you can develop tools to help you do it. For example, you could take sequences of DNA and assign them to boxes or parts, right? And you go, well, this promoter sequence is really part number 32, right? And okay, now all of a sudden we have a collection of parts, and you'd have to memorize the parts if you wanted to program in DNA, not the sequences themselves. But then you ask, well, maybe we could put the parts together to make still more powerful objects. Like I could take standard biological parts that each encodes a different biological function and perhaps organize them into higher level objects, which will make up a name. We'll call them devices. Now all of a sudden I could have devices which implement Boolean logic, and, or, not. I could have senders and receivers, transmitters, actuators, things that other types of engineers might be familiar with, right? And as we go from the low-level DNA sequence to parts and then devices, we can put the devices together to make systems. And what we are assembling here is a human construct. It's an abstraction hierarchy such that the person at the top, not that they're better than the person down below, but the person at the top who's working on a system, it could be a genetic system, and they're putting together devices from the next level down, but they don't need to know anything about DNA. They don't need to know that DNA is made up of 4 bases, let alone anything about how to assemble DNA and whatnot.
Finally, these parts need to be standardized.
You could have standards that define how pieces of DNA are assembled, one to the next, to make higher level objects. In the same way that nuts and bolts are manufactured in accordance with a particular standard for screw threads, 60-degree angles squared off at the top at the United States, what this leads to is that machinists can manufacture nuts and bolts at many different locations, yet we can all go to the hardware store and the nuts and bolts from these different machine shops are consistent with one another. With the BioBrick standard for standard biological parts assembly is a first example of a standard that's been widely adopted supporting reliable physical composition or snapping together of BioBrick parts or pieces of DNA.
Using these tools, synthetic biologists intend to tackle the still mysterious workings of the cell by starting from the bottom up, streamlining this complex network into workable parts.
People are just beginning to get a glimpse of that complexity. Take human DNA. Roughly 1 to 2% of the sequence of DNA is used in protein synthesis. 1 to 2%. So what's the rest of the DNA doing? Nature would have evolved this system with all this, leaving 98% of the DNA as junk. Well, no.
Yeah, biology may be too complicated to ever engineer reliably, but we're never going to find out the answer to that question unless we try and do it.
He's making it up as he goes along, which is as it should be. So we can manipulate the DNA, but not necessarily predict the consequences of that manipulation. Are we talking about biology, or are we talking about engineering?
Maybe we could just learn, as we become better at engineering biology, how to relate to biology as it exists in nature.
Synthetic biology is a little bit like abstract art. It's saying, we're not just observing the world. We're not just saying, look at this cellular mechanism and look at this event in nature. What the synthetic biologists are saying is, how can we recombine, how can we deconstruct nature and recombine nature in other ways that we might find interesting or useful? Problems that can't be solved with 19th century technology or even 20th century technology, how could 21st century technology solve them? Energy, clean water, infectious diseases that have proven intractable.
Synthetic biologists are at work already trying to tackle these pressing problems. The first synthetic bacterial genome, developed at the J. Craig Venter Institute, has caught headlines most recently. So too have microbes engineered to produce the malaria drug artemisinin, as well as others engineered to produce biofuels.
Bioenergy. Can you figure out how to rewire microbes to convert different biomasses into energy sources of interest? So say corn husks into ethanol. Or how can you create photosynthetic bacteria? And there's a huge set of opportunities to really get after real applications here.
I would like to write genetic programs that involve many different basic biological functions, right? By many, I mean, well, do I mean 10? Or do I mean 100? Or do I mean 1,000? Or do I mean 10,000 different biobrick parts? I probably mean 10,000 or 100,000, in the same way that microprocessors have many, many different components inside them. As a very different world than what we live in today with modern biotechnology, meaning biotechnology of the year 2008, where most of the systems being constructed involve very small numbers of components.
At this early stage in the development of synthetic biology, realizing its potential means laying a framework of openness and collaboration, an approach as bold as the science itself.
We set sail on this new sea because there is new knowledge to be gained and new rights to be won, and they must be won and used for the progress of all people.
It's important to recognize that synthetic biology grows up in a biotechnology landscape that's very far from open. It's largely closed, especially with respect to industry and commercialization. The way I think about it is much more akin to software development and computer programming. If you own the if-then statement, and I own the print statement, and somebody else owned the left parentheses, and somebody else owned the right parentheses, and every time we wanted to write a new computer program, we had to go talk to each of us individually and strike a deal. Pretty soon the cost of coming to an agreement and the licensing and legal terms would be much higher than actually doing the work, the research and the development of the technology and delivering the product. If you're sharing the components, if you're open about what does what, people will be able to make more powerful biological systems, right? Thus we'll have better biotechnology.
I went to parts.mit.edu, an open-source library of genetic parts spearheaded by Drew Endy and his colleagues at MIT.
The registry itself is two things. It's a computer database available via the internet. So if you go to the website, parts.mit.edu, you'll be able to access the database and download the descriptions of about 2,000 different biological parts. It's also a physical collection. So as people define and develop standard biological parts and then make them available for other people to use, the registry collects the DNA.
There is this commitment to openness and to open source and to the Wikipedia method of ideas. And that's an extremely interesting Jacksonian democracy model that's not really been tried. And let's do it across the world, and let's engage our youngest researchers.
Come November, iGEM teams will converge on MIT in order to show off much more than red bacteria.
Good Saturday morning to you, it is Angel Woodweir.
So I headed off to Cambridge, Massachusetts for the 3rd annual International Genetically Engineered Machines competition. And I arrived just in time for the first team's entrance. "54 teams of undergraduates from 19 different countries all came to show off their accomplishments in synthetic biology.
Students at schools all over the world are spending their summer designing biological systems, and then they all come to MIT for the Jamboree in November and show us the clever things they've built.
iGEM is about defining the synthetic biology community. Before you had iGEM, you had maybe a dozen researchers working on projects they would define as synthetic biology. Now you have over 400 undergraduates all speaking about the same thing, synthetic biology.
Okay, synthetic biology is the standardization of genetic engineering.
So I think synthetic biology is the true engineering of biology.
Standardization, modularization, and abstraction.
La standardisation, l'abstraction
You know, not necessarily bettering nature for nature, but bettering nature for us.
Anything that your mind can come up with, if you can find the parts for it in existing systems, there's no reason you can't cut it out, put it together, and make whatever the hell you want.
This cross-generational and cross-disciplinary exchange is unique to synthetic biology.
This summer we created a lead detector. So basically we have a cell that you can add lead to and it'll give us some kind of fluorescent output. The really cool thing about our system though is that it has an amplification component to it. So you can add lead to the cell will communicate that message to all of the surrounding cells, and then all of the cells will start to glow green. So we're hoping that we can eventually take this system and go out to the field and use it to test for lead in water or paint or anywhere.
And so what we did was we attached the cell to a polystyrene sheet, just a plastic sheet, and expressed a plastic binding peptide on its exterior. And so not only was the cell bound to the sheet, but then when you put the sheet in the water with the cell bound on it, it would fluoresce red if there was mercury present, and then it would bind up the mercury as well, and then you could pull the sheet out, and along with it would come the mercury.
After sharing ideas and biobrick parts on the registry's website, these young scientists embrace the opportunity to continue this dialogue in person.
So did you guys see the UCSF presentation?
Did they already do it? Oh man, was it good?
I just heard about it. That's what I was thinking about. Scott Moore told me it was the most fantastic thing he's ever seen.
Really?
Like, period.
He clearly didn't see the Cambridge presentation.
And I was like, you know it was all high school students, right? And he was like, no.
And it's wonderful now that even with our very simple system, we now have connections from Mexico and Bologna and Germany. There's people keep talking to us saying we have the same system, let's talk, let's collaborate.
All of iGEM lives on the internet. This is the only way it's possible for us to have such a diverse, geographically spread out group and still have things work very well and very quickly.
You know, so for example, here would be some packets from the registry. This is now 3 years old. It's a foil plate with 384 wells inside it, and each of these wells has a different piece of DNA. The specific piece of DNA in any particular well is a different biobrick part. So you would add a little drop of water to one of these wells, suck it up with a little straw or pipette tip, and off you go. Put it into cells and see what it does, or combine it with other parts and see what the composite system does.
So what's nice about this is the way we designed it with the different devices and different modules is that you can just kind of plug and play your controller into something else. Like, for example, where Austin mentioned the tumor-killing bacteria. So you could use the same exact chassis, and you could use the control if you wanted to, but instead of an iron promoter, you could have some sort of a detector that you're near a cancer cell, and then you could turn on a lot of production of maybe some sort of an invasive protein or something that would enable the bacteria to invade your cancer cell. So these other parts are useful in many other synthetic biology applications, and we've put them onto the registry in hopes that future researchers will be able to use them.
If you look at most projects here, most of them were half finished. But they have some really great ideas, that's for sure.
They tried lots of, I wasn't in that part of the project, but they tried lots of different combinations of things.
Oh, that's too bad.
And nothing, well, I mean, things began to work out right at the end, but then we didn't have enough time to finish off properly.
It's like it normally is.
Yes.
Always run out of time.
Yes.
Synthetic biology is new. Does it really work? Will it work well? And iGEM provides examples where actual undergraduates in a short period of time, by using standard interchangeable parts, are able to build interesting systems in one summer.
We can actually predictably engineer biological systems. We have to integrate the different parts, and so far we show that integration does work, as we have integrated 2 parts of our project. And we're working on bringing in the 3rd part too.
In short, it's a proof that synthetic biology can work, and it's doable.
And now there are 6 finalists. Finalist number 1! There it is! Finalist number 3, UK. Finalist number 4, Slovenia. Finalist number 5.
winner of the iGEM for 2007 from these 6 finalists is Peking.
These undergraduates are contributing members of the burgeoning synthetic biology community, demonstrating in one summer's work the efficacy of building systems from standardized genetic parts, and adding their own to MIT's registry. Here is a new generation of scientists.
I feel like contributing something back to the scientific community, which has trained me so well over these few years, and which has certainly given me a fun time over summer.
We think we have so much to offer, and we also have a lot to learn from other teams, that just the interaction, being able to share all these ideas and experiences just enriched us all a little bit more.
Next year there will be even more teams, fresh faces with new parts to work with.
No man can fully grasp how far and how fast we have come. This is a breathtaking pace, and such a pace cannot help but create new ills as it dispels old.
To better understand the potential risks of synthetic biology, I traveled to Washington, D.C. to speak to a distinguished security expert. Terrence Taylor is a former UN weapons inspector and currently heads the International Council for the Life Sciences.
When you have a technology like synthetic biology, where you could redesign or synthetically produce a virus that could potentially kill large numbers of people, you have to be very alert to that. So we're talking about something in terms of biological risks of a very high order. If things go wrong or somebody deliberately misuses the science or technology, which can kill as many people as a Hiroshima-sized nuclear weapon, and maybe even more if we don't get things right.
Synthetic biology has to take seriously that it is coming of age after 9/11. The idea of terrorists using powerful but simply learned mechanisms for manipulating viruses is a pretty terrifying idea. And it plays into American fears of savage others with powerful tools coming to threaten us. And so synthetic biology's moment of inquiry overlaps a moment of tremendous fear of the new in American life.
What we have to do, and this is so important in the approach, is to safeguard the advance of this technology. That's a very different approach than restrict, control, prevent. That is not the way to do it.
We are fiddling around. We know how to construct, we know how to put together sequences of DNA, but we don't know how to regulate the consequences of that construction, that synthesis, if you will, in the larger world. And we need to think about that.
And the question is, what do you do when you have the knowledge? What about application? But I think it's folly to suggest we shouldn't learn to know everything. I think we should think about how we apply things, and that should be the step in which we do our tightest regulation.
And if we wait too long, it'll be too late to influence the industry and academia to make sure they're promoting the best practices. You can do this through networking and promoting best practices. It already happens in other aspects of biosafety and biosecurity. There are There are biosafety associations in many countries around the world. So any approach to try to reduce the security risks has to be international. So I think it's a particularly challenging problem, but it's doable.
What's my biggest fear for synthetic biology? That people fail to grasp how quickly the technology is developing and how great a chance it has to impact what it means to be a human being.
So the more synthetic biology says, how is the world put together by deconstructing the world, the more it challenges our idea of who we are.
Partly to be provocative, my view is that life is a human category. That where we place the divide between the living and the non-living is a consequence of our conventions.
Everything that you know may not be true, and we have to find out if what we think is true is true. And that's a very destabilizing idea. And science is scary because it asks us to travel with scientists into a place that has not yet been discovered. And that is terrifying. Modernity may not turn out to be a safe place.
Remember in 1995, that's when the first bacterial genome was sequenced, and it was only 6 years later that we did a draft sequence of the human being, and it's only 6 years later, this year, that we just announced the 1,000 human being genome sequencing project. That's an example of an exponential technology.
With science and technology moving at such an exhausting pace, where will we be tomorrow?
Looking ahead in any science, and perhaps particularly in the life sciences, is a huge challenge. And if you were standing in 1985 and you asked the question, where is the information technology going to be by 1995, you would have been way off the map.
You end up having the ethics not of science, but of science fiction. And that's a pitfall, I think, that many people have fallen into, discussing, say, Jurassic Park instead of the actual things that's happening, say, in an IGM competition. We can't know the future, and that's what makes science interesting and fascinating. You can't know the end of the book. You can't turn to the end of the book and say, oh, this is how it turns out. It's science because it's experimental, and you can't know what will happen. That's the mystery, and that's the ethical difficulty.
Is it computer science that we draw upon? Yes. But it's also mechanical engineering, and structural engineering, and electrical engineering, and environmental engineering, and chemical engineering, and the history of technology, right? Architecture, urban planning. One of the most exciting things to think about are the lessons yet to be learned that haven't been encountered in other engineering disciplines. And so not only are we looking backwards to every other, sister discipline in engineering and the sciences, but we also can look forward to changing how some of those fields act by the lessons that might come forward as we get better at engineering the living organisms we care about.
The vast stretches of the unknown and the unanswered and the unfinished still far outstrip our collective comprehension.